All right. Hi, everybody. I hope that you are all doing well. So a couple of general announcements before I get into the material. On Monday, we're going to talk about a paper that talks about, it talks about some germinal center related stuff. But you will see that it does pull together quite a few things that we've been talking about recently. It's also a really interesting paper. I think it's a really cool paper. That paper is posted along with some questions about it. So that's Kinetics 2020. You also have I also posted an assignment for next Friday. Again, it's basically you read a paper and answer some questions about it. It's a paper that I think is really important in terms of us thinking about immunology big picture, and we can think more about that a little bit next week. In terms of things that immunologists need to really remember in designing experiments. With that one, I gave you the paper. I also gave you 2 short news articles, like talking about it, that may sort of be particularly digestible and helpful for you. So all of that stuff is on Moodle and ready to go, and I am in the most intense grading jail this weekend. So hopefully I will have other stuff for you on Monday. Today we're going to talk a little bit about some other aspects of peripheral B cells, just to sort of finish up. And then we're going to kind of think about aspects of kinetics and memory of adaptive immune responses. Yet another thing I adore talking about. This is one that is there are a lot of parts of this that are very much stuff I was doing in graduate school and near and dear to my heart. And so lots of good things I can talk about here. Just as a reminder, last time we were really focusing on B cell responses in the periphery. And specifically, we were looking at what happens when those B cells first are activated in the secondary lymphoid organ. So sort of this thing that's shown as sort of this phase 5. And we're sort of moving into what's happening with their differentiation. And when we were thinking about those B cells getting activated, you can recall that we talked about the importance of a T follicular helper cell in helping to activate that B cell along with antigen. And then this process of the germinal center reaction, where we're going to have clonal expansion of our B cell, but we're also going to have somatic hypermutation, as well as class switch recombination that are going to happen so that we can eventually get our affinity matured antibodies and our class switch. And so this is just a reminder about affinity maturation. So of course, we have point mutations that occur in the part of the gene that encodes the heavy and light chain variable regions. So the VJ part of the light chain, the VDJ part of the heavy chain, gets a bunch of point mutations. Here you can see these little black lines are showing point mutations in that region. And this will influence the protein, changing the affinity of that protein, the antibody protein for its antigen. And so we have mutation of the DNA, somatic hypermutation, that changes the affinity of the antibody. This is done, if you recall last time, by this enzyme AID. And AID is acting on cytosines in this region of the antibody gene, changing them to uracils. And then that's leading to point mutations where we get other base pairs put in and we change the base pair. And so that's how we're getting these point mutations. This deamination of the cytosines into uracil is happening in the variable region section of the heavy and light chain. It's giving us this change in affinity. We also are seeing class switch happening as a result of activation of that B cell in the germinal center. And so we have all of our different heavy chain region gene segments downstream of VDJ. Each of them has a switch site in front of them, and for class switch recombination, this same enzyme, AID, is mutating the switch sites, so it's deaminating cytosines in the switch sites to uracils, and it's basically leading to damage, DNA damage of that switch site. That switch site has a lot of Cs in it that all get changed to Us, so it's a mess. And as a result, the cell tries to repair that switch site by recombining it with another one, by doing homologous recombination as a part of DNA repair. And so in the end, that sort of damaged IgM switch site will recombine with some other switch site. We will delete the DNA in the middle, and we will now have this other constant region gene right downstream of VDJ. So we've basically had that B cell class switch. So again, notice that both of these changes are changes to the DNA. So they're not reversible. They are happening with this enzyme AID in the periphery. This is not RAG. RAG is never coming on again in this B cell, so it's a whole other process. But it is, in fact, a process that's happening that involves DNA change. One other thing that I didn't get a chance to mention last time as I was running out of time is that we had talked a little bit about hyper-IgM syndrome, and I told you that there were 3 genes that could be mutated to lead to hyper-IgM syndrome in patients. One of those genes was CD40. One of those genes was CD40 ligand, so the protein on the B cell and the protein on the T cell that interact with each other to allow the T cell to do the help. So if you can't get the T cell help, you can't get any of this process to work. The other gene that leads to hyper-IgM syndrome, or the mutation, is AID. So if you don't have AID, you also don't have class switch and affinity maturation. Just sort of makes sense because it's the enzyme that does those processes. And so we've basically sort of seen the outcome of this whole process with class switching and affinity maturation as a result of what we see in the germinal center, or as we learned last time, this. So first our T cell, it got turned on by a dendritic cell. It becomes a T follicular helper cell. The T follicular helper cell goes to the border where the activated B cell is, presents antigen. The T cell proliferates, but it also starts to have that B cell proliferate and move to the germinal center. The T cell also moved to the germinal center. Here, look, we're in the germinal center. There's the B cell, there's the T cell. The B cell is going to proliferate and mutate. Then it's going to go back and test with the FDC and the T cell again. Maybe it is happily ever after and it leaves. Maybe it goes around and around again. And so you can see that that B cell eventually is going to possibly be heading out of the germinal center after this whole process. Maybe that's the end of, and the T cell is also going to head out. And you can see this is sort of another view of that here as well. So here's the T cell getting activated. The B cell sort of gets activated to start with. The B cell then comes to this junction point with the T follicular helper. The B cell divides, makes a lot of copies of itself, starts mutating, tests with the T cell and the FDC to try to make more mutations and to class switch. Eventually, it will be ready to leave and go out and be our amazing antibody producing cell. So this is sort of the overview of what we saw last time. But there's one other thing that's described here on this slide, and it really is sort of on the border between what I wanted to talk about last time and what I want to talk about this time. So it's like perfect segue. Because just like with, if you remember, we talked about T cells getting activated, and we talked about differentiation of those T cells. So those T cells kind of gained a new function. They grew up. If you remember, our CD8 T cell became a CTL. our CD4 T cell became a helper T cell. And just like that, we can think about ways that the B cell could be differentiating and sort of gaining this new function, sort of the grown-up B cell name, just like we had the grown-up T cell names. And as you will see today, a little bit of what I was telling you about sort of the grown-up T cell was a little bit of oversimplification, but we're going to see some more details on that today. And this slide also shows you some of the results of the differentiation of that B cell. And so what you can see is that sometimes we get a B cell that's called a memory cell at the end of this, and we'll talk more about memory cells a little bit later today. Sometimes we get this cell called a plasma cell. And a plasma cell is sort of the CTL version of a B cell. It's the activated B cell you would call a plasma cell instead of just a naive B cell. And so a plasma cell is super, super highly specialized to make huge amounts of antibody. So if you want antibodies to be produced, if you want to make antibodies to something, you need to tell your B cells to turn into plasma cells, because plasma cells are basically antibody secretion factories. And you can see the difference between, so you can see some information about plasma cells here. This sort of shows you that B cell could become a plasma cell or a memory B cell. I'm going to say more about memory cells as we go forward, although I will, full disclosure, much of what I'm going to say about memory today is going to be about memory T cells, because, you know, there's that whole dissertation thing on that. But a lot of the things I'm going to say about memory T cells also can be related to memory B cells. Or we have this plasma cell. You can actually see an image of a plasma cell on the left. We also think a lot about locations sometimes with plasma cells. And so I'll say the most common location for a plasma cell is actually that it goes back and hangs out in the bone marrow. Though you can imagine it in a bunch of places. Technically, the official textbook thing is that we say plasma cells are in the bone marrow. So they go back to the bone marrow and just hang out there and just make boatloads of antibody. So I want you to take a look at this microscopy image of our plasma cell that you see here. You will not see any other cell in the body that really looks like this under a microscope if you look at it. So as you look at it, what do you kind of start to notice in this image of this cell? Yeah, David.
The nucleus is huge, and there's also every single area is rough ER.
Right. So if you think about like a normal cartoon version of a cell, you see a nucleus, you see a little bit of ER, and you see like the rest of the cytoplasm, right? You see all the other cytoplasm, you see all the other organelles. Can you find really much of any other organelles here other than ER? No. This cell, it basically is like taken over by rough ER. Rough ER is really important for making secreted proteins, like an antibody. So this cell basically gets rid of many of its other parts of the cell and just has the things it needs to make massive amounts of protein secretion. And so, in fact, this cell loses a lot of abilities. It gains the ability to make crazy high amounts of protein, crazy high amounts of antibody. But there are a lot of things it can't do because it doesn't have the parts of the cell anymore to do them. And so this is showing us the difference between a naive B cell and a plasma B cell. A naive B cell has immunoglobulin on its surface, aka it has B cell receptor on its surface. It has MHC class II on its surface. It's this naive cell. It hasn't really learned how to secrete much antibody. It may secrete a little, but it's not going to secrete a ton. It also can, grow, aka do division. It can proliferate and make more copies of itself. It can sort of survive well. It can do more somatic hypermutation if it gets activated. It can do isotype switch if it gets activated. Once the cell has become a plasma cell, it has one job and can do only that one job, which is making tons of antibody. It stops having B cell receptor on its surface, stops having class 2 on its surface, stops proliferating. It's not going to mutate more. It's not going to isotype switch more. It's basically like just focusing on this one job and has lost all other abilities. I have this one ridiculous t-shirt that has all the different immune cells as like little people, like with little sayings. And the plasma cell says something of the effect of like, I don't care. Because it can't be sick. You can't signal it anymore. It has no receptors. It lost those. It has just making boatloads of antibody. And that's awesome for you to make boatloads of antibody. We really like that. We're going to talk more about that a little bit in a second. And so this is kind of what we see if we look at antibody responses over time. No, sorry, Jazz, go for it.
I was going to ask, in the slide previous,
Mm-hmm.
you kind of see how it's been eccentric in the way that it's pushed to the side.
Mm-hmm.
Physically, like, what causes that physical displacement? Is it the amount roughly?
I think it's just the amount of, it's just trying to fit everything in there. I don't know if you would see that in all cells. That may just be an artifact of this cell. So we can think a little bit about what we're going to see in terms of antibody responses over time to an antigen. So of course, when we are looking at an antibody response over time with an antigen, we have a little bit of this phase where we're not making a lot of antibodies yet because we have not yet had our B cells differentiate into plasma cells. So that takes a little while, just like we saw with T cells kind of taking a little while. And at first, our B cells are probably making IgM because they haven't been activated enough to class switch. So maybe they're proliferating, they're starting to make some IgM, but they haven't actually switched yet. And so we get some good IgM production. And only a little bit later on do we really start to get IgG. So you can see a little bit of IgG here. This is showing you total. You can see IgM's most of the total, but there's a little bit of other stuff, especially as time goes on. So we get sort of IgG. Eventually, those antibody levels go down. We're going to talk more about the sort of that going down part of the process when we get to the T cells in a couple slides. But in fact, just notice here that that decrease is here as well. It's just I'm not going to talk about it as much here, but it's the same thing. But those cells that are going to remain are cells that have already gone through this process. They're going to be the memory cells that have already done all of this. And so those cells have largely already class switched. They've already done affinity maturation. They've already started to differentiate to be better at making antibodies. So if you trigger this person again, if you give this person antigen again, now you have a different type of cell responding. You have a cell that has experience, that has already class switched. It's already hypermutated. It's actually already learned how to make antibodies pretty well. And so you can see at this point, you get a way bigger amount of antibodies. So just the black line of total amount of antibodies is way bigger than the black line the first time. And you can see that it's mostly going to be IgG because we've class switched now, as opposed to the IgM that we see in the first time around. And so if you look kind of over time at a response to any particular antigen, this is showing you the responses over time. And here you're actually seeing first time seeing antigen on the actual axis. Here's the second time, here's the third time. You can see that we start out with a lag, IgM first, then IgG. Second time it's very IgG, and after that it's extremely IgG dominated. And you can also look, if we look at the affinity over time, M doesn't really get much better. The G's are getting better and better and better each time. And so this is part of the reason why, if we're testing someone to see if they're making a good antibody response to something, we often will just draw blood and see if they're making IgG. And so, because that's really telling you either you've gotten like this far in a response or you're at a secondary response. You're sort of seeing that microbe for the second time. And so we can kind of think about what antibody responses look like the first time you are infected with a particular microbe or the first time you see a particular antigen. When you started with a naive B cell that has to go through this whole process, or what you see when you're making a secondary antibody response, the second time you see a particular antigen. So one big difference is the first time when you're making that primary response, the B cell that responds to that antigen is pretty rare. That's not as many of that B cell. Later, the B cell is less rare because you've had a bit of proliferation, you've increased the number a bit. And so what you can see here is, this point that you're starting from at this second immunization is higher than the point that you were starting from here. So one difference is you're starting from a higher point. You have cells, these B cells are less rare. That second time around, you're really going to only have your class-switched antibodies being made. The first time, it's going to start with IgM, and only kind of a little, after some time, are you going to get some switch. The first time around, your antibodies are going to be pretty low. Second time around, your antibodies are going to be pretty high in terms of affinity. First time around, you're not going to have much mutation. You have to actually sequence the B cell, and you won't see many mutations in the sequence, which is why the affinity is so low. Later on, you'll have high affinity because the B cells have picked up a lot of mutations. So you can also see this here. So the first response is usually smaller. The second response is usually larger. The first response is usually IgM. The second response is mostly G. The first time around, you have this low affinity. The second time around, you have high affinity. So I have two sort of things I want us to think about with questions related to this. First question for you. Why is it when you got your COVID vaccine that you had to get boosters? Why did you have to get more than one shot of the COVID vaccine? Yeah?
To induce a larger amount of IgG. So
Yeah. You want to make sure that you are fully switched, that you have lots of G, that you have lots of antibody. You want to make sure that you actually have a primary and a secondary already done before you get infected. You want to make sure that you're getting this really nice secondary response. You want to have lots of antibodies in your body you don't want to just be here if you get infected. You want to be here with this super nice response. So you want to have gone through two of this process when you are getting that vaccine in order to really have the best possible protection. So some of what we're seeing here relates directly to why we do things like have booster immunization. Though we can talk more about that when we talk about vaccines later. Yeah, Emily?
So it actually doesn't wear off. It's just okay.
So much we can say. Some of that's going to come up later, but there's so much I can say about that.
Okay.
No, it's really about later stuff today. It's really an immunology problem. So the other thing that we need to think about is also shown on this slide, which is that primary response is something that we can see with all types of antigen. While secondary responses with nice class switch and somatic hypermutation, making memory cells, making plasma cells, making like boatloads of antibody, are only seen if the antigen is a protein. So we need to think about this a little tiny bit. Because in fact, when one develops a vaccine, this is a huge deal. And we can talk more about how we actually have to work on our vaccine strategies. Because there are some vaccines that you have received where the antigen that you need to make an antibody response to be protective is a sugar. You have received antibodies vaccines where we actually want you to make an immune response to a sugar, and we have to do all sorts of tricky engineering because of this issue. And we can talk more about that tricky engineering in a bit. So if we think back to exam 1, I told you that B cells can respond to nucleic acid or protein or lipid or carbohydrate. I told you that the antibodies can see any of those types of molecules. And you can see that is what I'm telling you here for the primary response. But then I'm telling you that the secondary response is only something that we see if the antigen is a protein. Why is that? What's up here? Yeah, Jazz?
I want to say it has to do with T cells because for T cells the antigen can only be a protein.
It's got something to do with T cells because for T cells the antigen is a protein. So what is going on here with T cells? What's up? Yeah, Paige.
Is it because the B cell interacted with the T cell?
Yeah, the B cell actually has to be able to present the antigen to a T cell and get T cell help. The only thing that B cells can show T cells are proteins. So if the B cell doesn't have the protein to show to the T cell to get T cell help, that B cell can never get T cell help, and that B cell can never undergo this whole process. And so this process that we see is something that only works if we have a protein antigen, or if we do some fun biotechnology types of experiments, which, again, we can talk about. So this is pretty key to be thinking about. But then the other part of what I want to just generally talk about is about general ideas of sort of the trajectory and kinetics of responses. You saw my example of trajectory and kinetics here. You can see what this looks like for B cell responses. You can also see that on this slide, I'm calling it the adaptive immune response, because really we see this is the same thing we see for B cells and T cells. I'm just going to go into depth with it on T cells, just as an example. But a lot of the exact same things I'm going to tell you about T cells apply to B cells as well. And there are a couple places where I'll give you the B cell example because it's particularly helpful. And so you can kind of see this curve, and you can see, when I made this curve as well, where we inject or we see the antigen for the first time, we have a little bit of a lag, and then we finally generate this really nice adaptive immune response a bit of time later. But we haven't really talked about what the rest of this process looks like. So kind of this is where we've been so far. We've had our naive T cell interact with an antigen presenting cell. We've had clonal expansion of that cell. We've had that cell differentiate. We've had the B cell be activated, do some clonal expansion, differentiate. So we've kind of seen this so far. And that's all really, really, really important because that is what's going to make us eliminate the antigen. That's what's going to make us get rid of the microbe. So you can see once we have differentiated those B cells into plasma cells, those T cells into either CTLs or helper T cells, we're now going to eliminate the antigen. And you can also kind of see this here. So the microbe is going to sort of have free reign for a while the adaptive immune system isn't doing anything. Eventually, we're going to get this nice adaptive immune response, and it's going to get rid of the microbe. And then you're going to get better. And you can see this here. So here is the amount of virus in black. You can see that there's a lot of virus replicating. We finally have the CD4s and CD8 responses come up, and that gets rid of the virus. So the result of this immune response is you get rid of the microbe. Yay. And part of the reason why you're getting rid of the microbe is because you have these lymphocytes that have undergone differentiation that are now really good at killing or at dealing with microbes. So this is, again, our CTL or our helper T cell or our plasma cell. We often refer to all of these cell types in general this sort of activated lymphocyte as an effector cell. So we basically have our naive cell that's going to become a cell type called an effector cell that we've seen here. So, we've seen this change into the effector cell. And you've already seen this in the case of T cells, that naive cells and effector cells are different from each other. There is something that happens to the cell as a result of this experience and this differentiation. One thing that you've already seen is that the naive T cells required signal 1 and signal 2 to be activated, while the effector T cell only required signal 1 to further sort of do its function. So if you wanted to give a signal to a naive T cell, you need signal 1 and signal 2. If you want to sort of give a TCR signal to an activated T cell, you only need signal 1. So there's one difference between a naive and an effector T cell. Earlier today, we mentioned a difference between a naive B cell and an effector B cell, the plasma cell, which is that the plasma cell has changed its cell structure a lot. It's mostly full of rough ER, got rid of lots of other stuff, doesn't even have a receptor on its surface anymore. It's just going to be an antibody production factory. So that's the effector cell versus the naive cell. There are lots of other ways that naive cells and effector cells differ from one another. So one of the differences is how much they will make of different products, like how much they will transcribe different products. So for example, here's one product, IL-2. An effector cell makes 1,000 times as much as a naive cell. So we've got all sorts of different products where effector cells are just going to be able to make so much more of the product than the naive cell. And you can also, of course, have seen that before with the antibody, that the effector cell is making so much more than the naive cell. We also see differences in a bunch of different cell surface proteins, so we're able to distinguish naive and effector cells by flow cytometry. There are differences in the trafficking. So remember we talked about how naive T cells want to go back to the lymph nodes, whereas effector cells want to go to tissues. They've changed what kind of trafficking proteins are on their surface. So in fact, that's on here. So like there's some chemokine receptors and there's changes on their surface, changes in things like interferon gamma production or granzyme B production. Changes in, so here's some more trafficking proteins, here's some other types of proteins. So it's a big change in the effector cell. And if you look at these changes are all really, key important changes. One really hot area of immunology that I will just mention to you that people are now into is people have actually realized that these cells do different forms of metabolism to make their ATP. So there's this whole field of immunometabolism. In terms of whether cells are doing things like oxidative phosphorylation, glycolysis, or fatty acid oxidation. And so you can see here's basically a naive cell is doing this blue one of oxidative phosphorylation and the yellow of fatty acid oxidation. And you can see it switches as an effector cell to be doing instead some oxphos and some glycolysis. And in fact, you can even see with our different subsets of T cells, we have different metabolic processes as well. So you can see these naive cells and effector cells are very different from each other. It's like a whole different program they are doing. And if we think about this whole different program, it's great. It's awesome. We're excited. We're getting rid of our microbes. This is all fabulous stuff that we're seeing. But there's one other part of an effector cell that is important to think about. When you think about, say, that plasma cell I showed you, when you think about a CTL that is making all these granules of cytotoxic stuff, when you think of this cell that's making 1,000 times as much IL-2, et cetera, think about if I suddenly said, Emily, I need you to make 1,000 times as many experiments as you have been doing in the lab. Let's imagine I said that to you. How would you feel if I said that to you?
I don't have the time for that.
Okay, so you're like, I don't have the time. But let's imagine you had to do it. Just like the effector cell has to do it, huh?
I don't have like the physical capabilities.
So you'd be like worried about your physical capabilities. You'd be worried about your time. What would you probably have to do to make that happen?
Hire people.
Okay, what would you do, Jazz?
Or half-ass it.
Okay. So what if you're not going to hire people and you're not going to half-ass it? What else could you do? Huh?
Work 24/7.
Okay, you're going to work 24/7. What else? So you're going to work 24/7. What else might you do? Are you going to like study for this class and do the homework for this class? No. You're going to do the homework for any of your classes? You're going to like stop doing a lot of other stuff so that you can focus on doing just that, right? You're going to be like, can't do all that other stuff. I just got to work 24/7 on this one thing, and even that is going to be rough, okay? So you make this change, just like the plasma cell starts making all those antibodies. Basically, as you see, it stops doing the homework for everything else. It just has rough ER. It stops doing everything else other than making antibodies. We can see all of this. So, Emily, I ask you to do this. Let's imagine you successfully do 1,000 times more experiments.
Okay.
How are you going to feel?
Exhausted.
You are going to be exhausted, right? And so the good part about an effector cell is that an effector cell is amazingly good at doing a function and getting rid of some microbes. The bad part about an effector cell is in getting this kind of specialization, it also has had to lose out on some things. It's had to make some compromises. And so when we look at effector cells, one other thing that we notice is that effector cells, say, stop making proteins like BCL2. Anybody remember BCL2 from Bio 250? So you can see we go from a good amount of BCL2 to like none. Yeah.
Wait, it's in the song Nadia. It regulates apoptosis.
It does. What does it try to do when it regulates apoptosis?
Backs and bad?
It does. What's BCL2's point? Like, what's its goal? What is it important for? Yes, it combats backs and bad, but that's not what that's more detail than we need right now. Yeah, Roxy.
Doesn't it block the program death?
Yeah, it keeps the cell alive. It's the survival thing. BCL2 is the main thing that keeps that tells the cell you should be surviving right now. Effector cells ain't got time for that. They don't make any more BCL2. And so effector cells tend to be very short-lived cells. They don't tend to live a very long time. And part of it's because they have like gone so hard in their specialization that they're not going to be able to live for a super long time. In fact, there's one scientist in some of this literature who actually often refers to these cells as SLEX or SLEX, which stands for short-lived effector cell, because the idea is that effector cell is going to live for such a short amount of time. So we have our sort of naive cell that becomes an effector cell, but that effector cell is eventually going to die now because it has sort of gone so hard as being an effector cell. And I've already sort of mentioned a little bit about this as well, because I also told you that when we have our effector T cells, when we activate our T cells, I told you that they start making CTLA-4. And they basically get ready to get turned off. So whenever you turn on an immune response, there's already sort of this programming of this immune response is not going to last forever, you got to turn it off. Part of that's CTLA-4, part of that's just the fact that the effector cells are so short-lived. And so kind of to address Emily's question, we have to think about what happens sort of as our immune response goes on. And so after we have this clonal expansion, we make our clone army, we make these super differentiated cells, both actively through things like CTLA-4 and some others that I'll show you in a second, and kind of passively just because these cells have like gone hard and are now really short-lived cells. After the antigen is eliminated, this immune response is going to contract. And so most of those cells are going to die. Most of those cells that we have turned on, we have sort of primed up and gotten ready are going to they're now effector cells, and the effector cells are not sticking around very long. They are going to die. And only some cells are going to survive to the end of this process, and these are going to be the special group of cells, the memory cells. So basically, you're going to start with naive cells. You end up with sort of, this is a point of such debate in the field. I'm going to give you an answer, but people would fight me for it. You have sort of some effectors and some memory cells here. Like you turn them on, they all go from naive to either effector or memory. The effectors die, and only the memories are left after this process. And so this is this process known as the contraction phase of the immune response. And the contraction phase is actually extremely important. When I was in grad school, we were working on vaccine development. And we wanted to have our animals get bigger immune responses to our vaccine. And so one of the things one of my fellow students and I said was, we're just going to block contraction. We're going to just set it up so that contraction never happens, and it's going to stay big. And then we aren't going to have to worry about fancy ways to like make this go higher. We're just going to let it sort of do its thing. And one of the things that we learned the hard way is that it's basically impossible to block contraction. Contraction is so incredibly important and is basically sort of programmed into the cell when that cell is activated. You can't sort of not have contraction. And so here you can sort of see this as well. Whenever we're drawing our primary immune response, we always have to see a contraction phase after antigen has been eliminated. You can see that in this image as well. Yeah, we get this nice amount of CD4 and CD8 T cells that got rid of our virus, but then after the virus is gone, we contract our CD4 and CD8 T cells. So here you can see our effector cells expanding, and you can see like 95% of those cells are going to contract. So you can see the vast majority are going to be contracting. We often refer to this process of cell death and the way that these cells die as activation-induced cell death, or AICD. That's why that enzyme we learned last time that we wanted to call it AICD couldn't be called AICD because this was already named. And so once our T cells are activated, they are basically programmed to die a little while later. Some of this is due to things like CTLA-4. Some of this is due to cytokines. Some of this is due to a lot of different things. But basically the idea is that once we activate a T cell, unless that T cell becomes a memory cell, it's on a short time clock. It's going to die as a short-lived effector cells. Only the ones that become memory cells are going to live for a longer time. One of the proteins that is also really important here is fast ligand again. Last time when I told you about fast ligand before, I told you about activated CD8 T cells having FAS on their surface so that they could kill other cells having fast ligand on their surface so they could kill other cells with Fas. So you can see this interaction. You can see this cell undergoing cell death because of the Fas ligand on the CD8 cell interacting with the Fas on the infected cell. It turns out once you have a activated T cell, the T cell also starts making Fas so that it can get killed easily. And in fact, sometimes CD8 T cells kind of like curl around on themselves and kill themselves with Fas ligand interactions. And so basically when we are activating these cells, we're also kind of putting them on this timer when they are effector cells to live for a sort of short period of time. So we got to think about this a little bit. And there are a couple things we can think about with this. And so one of them is, well, I've told you that this contraction phase is super important. You know, we can't not have this contraction phase. And so let's think a little bit about what it would be like if there was no contraction phase. What would happen? Why would that be a problem if there was no contraction phase? And there are a couple reasons why this would be why it would be a problem if we had no contraction phase. And some of these exact problems, well, some of it we can kind of talk about theoretically, but there actually are patients who don't have contraction phase. So there actually are patients with a mutation. So I don't know why my fellow grad student and I didn't think about this, why we were kind of not like, oh wait, there's a disease that's really bad that happens if you don't have contraction. And so there is a disease called ALPS, or autoimmune. ALPS stands for autoimmune lymphoproliferative syndrome. So patients with ALPS, one of the things about them is they actually have giant swollen lymph nodes that you can just like see normally on a random day walking around. It's hard, the way the lighting is here, it's hard to see, but like here is a lymph node in this little girl that's just there all the time. She's got another one here. She's got the lymph nodes under in her right here. Super swollen. Her lymph nodes are basically swollen all the time because her T cells are never, her cells are never contracting. ALPS happens because of either a mutation in Fas or Fas ligand. So you can't have that death that I just showed you on the previous slide. And patients with ALPS tend to suffer from multiple different autoimmune diseases. So we see pretty massive autoimmunity and sort of general inflammation and sort of excess inflammation in these patients. So what's one reason why contraction is super important? What do you sort of see from this already? Why would you imagine contraction is important? Yeah, Roxy.
Because it helps control inflammation.
Yeah. Remember how we talked about the fact that the effector cell, it doesn't need signal 2 anymore, right? It's kind of like this, dangerous rebel that's going off and attacking whatever it wants now. Its sort of brakes are off a little bit. You don't want that around and active for a super long time. Sort of imagine it like you had a bunch of, think of those cells in some ways as a bunch of like 8th graders. It's one thing if you have a bunch of 8th graders and you give them an activity to do, right? It's another thing if you just let them loose with no activity to do and let them get up to whatever shenanigans they want to get into, right? So these T cells have to be short-lived when they're activated because we don't want them to live long enough to get into shenanigans when the virus is gone. We want them to just be dealing with the microbe, and then we don't want them around to do any autoimmunity shenanigans. We want them dead. And so if you actually leave those T cells on for too long, they get rid of the virus, but they're still active. They still don't need signal 2. Now they're like, what else can I kill? What else can I ruin? And they're going to now start leading to inflammatory and autoimmune processes. And so if you don't have contraction, you actually usually end up with pretty massive autoimmune issues. Even without that, though, there's sort of one other thing we can kind of think about and imagine. So I told you last time that if I looked right now at some of your lymph nodes, I would find germinal centers. You're making immune responses to stuff all the time. You are encountering microbes all the time during your life and making immune responses to them, okay? I can't even quantify, but you can try to imagine a big number. You can pick whatever big number you want of how many microbes you personally have responded to in your life so far, okay? And when we look at our response, you can see that, that when we had our expansion of those lymphocytes, we've made, 1,000 times as many, sometimes even 100,000 times as many as we started with for our clone army. What would happen if that happened to you and you didn't actually then lose some of those, you didn't contract or kill some of those cells? What would happen in your what would your lymph node be like? Yeah, Emily.
It'd be like a giant ATP sucker upper.
Be a giant ATP sucker upper, definitely. What else? Yeah, Miles. It'd be full. There'd be no more space, right? There probably also wouldn't be enough IL-2 to go around and stuff like that. Literally, if you did not contract, you have to contract an immune response so you have room for the next one. At some point, your lymph node would be out of space and you'd be like, sorry, no more responses, no space, no IL-2, we're full. At this point in your life, you would be a walking lymph node if you didn't contract any of those responses. You would literally have no other cells and no other ATP for anything else if you tried to keep every activated cell you ever made. And so it's actually really important that we have this contraction or this activation-induced cell death phase so that we keep our immune system ready to go and respond to the next thing that we might need to respond to. Yep, Jazz.
So, if you didn't have contraction, it's like the lymph node capacity almost, or you don't have enough?
Potentially, yeah. And so, technically, even in the ALPS patients, when they're missing fast and fast ligand, they have some contraction through other mechanisms. They've just lost one, and their lymph nodes are visible. So, if you lost all of the mechanisms of contraction, yeah, you would really just not have, you would just be a lymph node. So when we think about immune responses, this is really what we're thinking about in terms of what those responses look like in responding to a microbe. So we have this period of time, so we first have a very low adaptive immune response to our microbe because we just have naive cells and we have one for every epitope. If we actually are infected with a particular microbe, we go through all the steps of activation and proliferation and differentiation and trafficking that we've been talking about, and we eventually get this really nice high immune response, and we get rid of that microbe. And for some period of time, the immune response is still pretty high, definitely higher than it was. During that time, if we encounter the microbe again, we're basically going to get rid of it like that. That microbe is probably never going to be able to even get established and reproduce in our body at all. You can see it's listed here as an abortive infection, but whatever. So we have this period of time where you're really not going to get sick. You're really super well protected. But as time goes on, that response, as you can see, wanes. We get less and less of a much smaller response through the contraction phase, which is super important. But the cells that have lived through all of this, the cells that have made it through this whole process, are a different type of cell called a memory cell. They're not an effector cell, they're this type of memory cell. And the memory cells, again, have new properties that mean that when they see antigen again that second time, they are going to be able to respond really quickly. You can see that they don't have the delay that we saw the first time around with the naive cells. You can also see that the amount that they increase is way higher. And so we're going to have these memory cells that are going to act way better the second time around. This has been something I feel like I have had conversations about with people during COVID about so many times. And this is a thing that I think a lot of people have that have like made it to the New York Times, and sometimes I'm like, what are you guys doing? So Emily asked a question earlier about whether or not something wears off. Based on this, Emily, what do you think? Does your vaccine, so let's imagine we're not going to just forget boosters for right now. We'll think about boosters later. Just imagine, give you a vaccine. It's the only one you're going to need for your whole life. Does it wear off?
Yes, but not completely.
Okay, so Emily says yes, but not completely. Anybody else have any answers for this? Yeah, Miles.
Maybe like a very significant amount, but like not fully. Like sometimes you have to get like a vaccine and then 10 years later you have to get it again.
Okay, so you're kind of like maybe, so you're sort of all feeling like you're like maybe, I don't know what's going on. Yeah, Jazz.
My thought is that like after contraction, the cells that are left are memory cells.
Mm-hmm.
So all of those cells should theoretically just be there.
So they should, yeah. So here's the question. Here's actually what the real answer, the real thought is here. Is I would come back to you if you asked me that question, and I would say, what does wear off mean? Are we talking about how many antibodies you have? Are we talking about how many effector cells you have? Are we talking about how many memory cells you have? Does the amount of effector cells change over time? Does the number of effector cells go away? Yeah. If we think about B cells and plasma cells as our effector cells, so we are not going to have as many plasma cells over time after our vaccine. So is the amount of antibody, if I do a measurement of just antibody levels in your body, is that going to go down over time? Yeah, it should, right? Like that's the point. That like it doesn't work if that doesn't happen. The amount of antibodies are supposed to go down over time. But in theory, the memory cells should be sticking around, and you should, in fact, be really well protected. In a lot of experiments people were doing post-COVID when they were trying to look at how did the vaccine wear off and things like that, they were measuring level of antibody protein. They weren't measuring the amount of memory cells. Part of that's because it's really hard to measure the amount of memory cells, and it's relatively easy to just draw some blood and see how many antibodies are there. And so people were like, oh my gosh, it's weird, we're freaking out, the world is ending, ah! And part of me was like, yep, they went down. They're supposed to go down. Good job immune system doing what it's supposed to do. And so in fact, yeah, your immune response should go down by certain measures, while by other measures it might actually be better because it's this type of memory cell. And so this is one of many, 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 many examples where when people ask me about, well, what's going on with the immune response to this? I'm like, do you know how many parts the immune response has? You need to be way more specific in terms of what you're measuring. So there are bunches of more things I can say about this, but yeah, David.
Wait, so if memory cells are supposed to exist basically forever,
Yeah.
then what's the deal with rabies? And you have to get like an immune, like a booster shot every time you more or less get bitten?
So there are different levels of how well memory cells can persist based on how those memory cells are made. And so that will be part of our vaccine discussion and kind of how different vaccines set up different types of responses. Some of it also has to do with some of the unique features of how rabies works. The short answer I will say is that the vaccine is suboptimal. If we could make a perfect, an optimal vaccine, then that wouldn't need to be the case, but we can't really, we don't totally know how to do that. Yep.
So does that like kind of apply to the flu as well? Like the fact that you can get it every
So-
temperature and stuff like that?
So, there's so many, like, again, this is like my stuff. So like, there's so many things I could talk about for this for so long. Flu is pretty unique because flu has a mechanism of genetic change that other viruses don't have. And so flu is a different virus every year, whereas other viruses don't change at that same rate. And so the reason why you have to get a new flu shot every year is because the predominant flu strain that is circulating each year is different. And so you actually have to get a new flu vaccine. There are ideas and experiments people are doing to try to make a flu vaccine that could hit all strains, but we don't have, but because our current vaccine is really, really strain specific, we have to give you the vaccine for that particular year each year.
Yeah.
There is debate in the field about whether or not you need an updated COVID vaccine for the different genetic changes in COVID, or is actually any booster good enough? Versus does it have to be a specific booster? I actually think that any booster is good enough and that it doesn't have to be a specific booster. And some of that's due to some really interesting data that we've seen about how somatic hypermutation works over time. It actually looks like if you let somatic hypermutation go for a while, you get the ability to respond to variants through somatic hypermutation. And that's something that's only been discovered in the past like 5 years. And so I think that having more boosters is good, but I don't necessarily know that they have to be matched boosters at this point. I'll answer Jaz's question and then remind me I have to tell you about a study from Israel. But yes.
So when viruses like make variants,
Mhm.
pre-variants,
Mhm.
is it primarily changing the surface of the virus?
So, why would you think it's primarily changing the surface of the virus?
I think so. Well, more specifically, because that's what's being targeted by B cells. But to get in, we'd use T cells, though, I guess.
So
Both.
if the virus wants to change, if the virus makes changes on the surface, it's going to be hiding from antibodies. The virus might make changes in various places, it's going to be hiding from T cells. But everybody's T cell response is different, so it's really hard to hide at a population level. Everybody's antibody response is the same, so it does make more sense to try to avoid everybody's antibodies because you can't avoid everybody's T cells. So yes, because of that population level similarity of antibodies. No, because technically you can be trying to respond to each one. And so it's sort of an individual virus versus virus population would answer differently there. So in any case, people have sort of talked about, how long does the COVID vaccine last? And the answer is, what's last mean? Do you mean give you high levels of antibody in your blood? Do you mean give you high levels of memory cells that are going to act? People haven't actually done a great job at distinguishing between those two things. When the New York Times is like talking about whether or not your vaccine lasts, they're kind of just like hand waving about antibody numbers. The memory cell levels, I think people are still a little bit jury's out on them, but in fact, those memory cells look great. Memory cells actually, from what I've seen, look like some of the best memory cells we've seen. They're going to last for a super long time. Though, as you'll see in the paper we talk about for next week, there's some other questions we can think about with this. So there's actually a study in Israel that basically you're in this phase where you have this high amount of antibody and are absolutely like not getting infected again for about 7 weeks after each booster. Then after that, you could get infected again, but you're going to have a memory response and just have a really nice protective response. So are you trying to get protected against like that, even having the virus get into your body at all, or are you just protecting against having the virus do enough to make you sick? Is sort of this question that I think a lot of people are really thinking about and asking. And again, people are often not specific enough in how they are asking questions and thinking about this. Yep, Julie.
Is there, I know you were kind of just answering this, but like, is there a specific amount of time that memory cells last for, or is that like depend on the virus?
So you guys are asked, so a lot of the questions you guys are asking are about what's up with the memory cells, right? So you can see some of the cells become these effector cells, but some fraction of them become memory cells. Part of this is because they have survival proteins, like I told you before. Part of this is about their ability to respond to cytokines. And they are, in fact, different than those effector cells. And so here you see some of the naive cells will grow up to become effector cells. Some will become memory cells. There might be interconversion between the two of them. Some parts of that are more controversial, and we're not going to have to focus on that. But I could go on and on again about that one. But so there are these memory cells. And Julie asked this interesting question of how long do the memory cells live? This is stuff that we're only now really being able to start to address. And I will show you some really cool answers. And they're really cool answers. Like, really cool. But I will also tell you there's one little thing to know about these answers. All of the answers I'm going to tell you about are from measurements in the blood, not measurements in the tissue. And so we can tell you how long we can see memory cells in the blood, but we don't often do some of these measurements of them by actually pulling somebody's bone marrow. We don't actually do measurements necessarily of looking in your spleen or in your lymph node or in some other tissue. Memory cells also often like to hang out in lymph nodes, just to like soak up the cytokines. And so one of the real issues with things like the COVID vaccine is people have been doing some experiments with memory cells in blood. We have not done any, nobody has like taken people who got the COVID vaccine and gone into their bone marrow to see how many long-lived plasmas, how many certain long-lived cells are in the bone. Like, so know that sort of, there's a lot that's still out there in terms of, well, where do the memory cells actually live? We measure them in the blood because it's easy to measure the blood. But in fact, they might be living in various places where they get lots of good cytokines. And so maybe we're underestimating. And so know that I would say the answers I'm going to give you, which are crazy cool answers, are underestimates. Yep, Jazz.
Okay, like this is a more niche question, but like patients who have multiple myeloma consistently and like frequently get bone marrow aspirations and samples taken because of like monitoring bone lesions and things like that. Individuals who have multiple myeloma are also immunocompromised because of the cancer. So would it be hard to do a test on their aspirations and looking at the memory cells because of it? Like, would it be like, I guess, a construed?
I think I would worry about whether or not their memory cells are really representative of healthy memory cells because of all the other things going on with them. But I don't know. So one other thing that's sort of interesting in answering Julie's question is you have to think a little bit about how the heck you answer this question of how long memory cells live. Because one of the things you have to think about is really to answer the question of how long memory cells live, you've got to think about knowing that, like, when did the microbe, you get the pathogen, and how long you've had it. And really what you want in that kind of experiment is you want a situation where somebody saw an antigen one time, made memory cells, and never saw the antigen ever again. And you knew the timing so you could see how long that lasted. That's pretty hard to do in people. But there is a situation where we have done it in people, where people have been able to do it in people. And the reason it has been able to be done in people is that there is a virus, which is smallpox virus, that has been eradicated from the earth. So the last case in people was in 1977. There have been no people on earth that have been infected with this virus since 1977. And they used to give everyone this vaccine. And so there are a bunch of people who got this vaccine and have never been infected with the virus. And the other thing you have to realize is that if you actually want to see how long memory cells last, you probably have to look in old people, right? And there are other maybe issues about whether old people are representative. And so that also is sort of a caveat to remember. But when they actually looked at really old people who got the smallpox vaccine and then never were infected with smallpox, they could figure out how long memory cells lived. And so they see that antibody levels don't, particularly if you're looking at memory cells, they never really found a decline in the memory cell. And CD4 and CD8 T cells seem to have a half-life of 8 to 15 years. They could detect memory cells in the vaccinated individuals at 85 years post-vaccination. So in this human study, the answer was 85 years at least. Someone also did a mouse study on this. It's such a cool study. Such a cool study. And one of the things that's cool about this mouse study, I thought about us reading this mouse study because this paper is amazing, is they actually they had mice, they gave them a virus, they had the T cells become memory cells, and then they took those memory cells from the mouse and put them into a new mouse. So they didn't have to worry about the elderliness. And they could also then let this go longer than the lifespan of one mouse. So they could move it from mouse to mouse to mouse to mouse, different generations. When the paper was published, the cells were still alive. So the answer is this is how long they got it to work. That doesn't mean that's the longest it could go. They're actually still going right now. So the answer is longer than this. Just so you know, just for your knowledge, the longest a mouse really can possibly live, and this is like the longest. This would be like an insane mouse, is 3 years. And that's like in the lab, we never really get them above 2. But like 3 years is the absolute longest a mouse can live. The cells are going up at about 12 years right now. These memory cells from the original mouse. So 4 times the lifespan of the animal. So Julie wanted to know how long memory cells live? A long time is the answer. And again, this is only looking at the ones in the blood. We haven't necessarily been able to always look at tissue. And abilities to actually measure memory cells are things that really were just getting figured out when I was a grad student. That's why it was such an exciting area to work on for my PhD. Yeah.
You're not exposing those memory cells again, like to test the longevity, then how do you know that they're still working effectively?
So in the mouse experiment, they did experiments and pulled them out and checked. In the smallpox longevity study, they basically pulled them out after a long period of time and again did the experiment, but you have to check. I will also say that, as you can see from all of this, I would say that there are some memory cells after this long period of time. It's not the number you had before. They are sort of going down with some half-life over time. They are, people talk about the waning of the immune response, and there is absolutely waning going on. And we can talk more about the waning at some later period of time. So know that it's not like you're staying at max awesome memory cellness. You are getting some waning in those memory cells. And the memory cells also have a lot of sort of interesting properties in terms of letting them survive differently, be stimulated differently, have a lower threshold for activation so that they make really huge responses. Differences in trafficking, all of those types of things. And so this is sort of why we see a secondary immune response, is because now after we've had our first primary response, these cells that remain are memory cells. Note that there are more of them than there were of the naive cells. So there is this increase in number. But these cells also are better at functioning. So when they are triggered, they respond really quickly. They're basically already differentiated, so they already can be effective against the microbe. They can divide really fast, function really fast. And as a result, you are able to sort of clear that microbe really quickly, probably before you actually start to see symptoms. Or in some cases, with only mild symptoms where the first time you had more severe symptoms. So you're gonna have a much less illness. You're probably gonna have less amount of microbe. You're gonna have microbe for less time. This is sort of the idea of why the idea is you can't get certain things twice. You actually can get it a second time. It's just that you're wiping it out before you have symptoms. You didn't know that you're having it twice. This was really easy to understand pre-COVID. It got all confusing during COVID because we actually found out really sensitive ways to test for, is the virus in your body at all? In the 60s, we would look for, did you get disease? And we'd be like, no, you didn't get disease. You never got the virus ever again. Now we do super sensitive PCR to see if you got the virus, and we find out, yeah, you might get it again. You just aren't as sick. It's much, less severe. So people talk about breakthrough infections, where there's a little bit of virus the second time around, not enough to make you sick, but a little bit of virus. And they're all like shocked with COVID and the breakthrough infections. I made this slide in 2009. They're supposed to, that's how it's supposed to work. This is sort of basic immunology and what it's supposed to look like. So we will talk about a paper that has some pretty cool stuff about B-cell responses. And next time, and I'll also be giving you some coronavirus-y background because it's relevant to the paper. And I will see you guys next week. Remember those discussion questions, and also that there is that other paper and assignment for Friday.